The overall goal of the following experiment is to visualize and quantify BMP signaling in tissue culture cells. BMPs bind to receptors in the cell surface and phosphorylate 3 SMAD effectors, SMAD1, SMAD5, and SMAD8. These form complexes with SMAD4 and translocate to the nucleus where they orchestrate transcription. Here we use PLA to visualize complexes forming between the endogenous SMAD effectors and SMAD4 in tissue culture cells with or without BMP stimulation. This begins with parallel use of 2 primary antibodies, one of which recognizes all 3 phosphorylated SMAD effectors and the other of which recognizes SMAD4. Secondary antibodies, called PLA probes, each with a unique short DNA strand attached to it, bind to the primary antibodies. Where the PLA probes are in close proximity, the DNA strands interact through a subsequent addition of 2 circle-forming DNA oligonucleotides. These are joined together by enzymatic ligation and form a circle that gets amplified via rolling circle replication. After the amplification reaction, hybridization of labeled complementary oligonucleotide probes highlights the product. Fluorescent spots associated with the amplification of each circularized DNA product are easily visible when viewed under a fluorescence microscope. The spots correspond to complexes between effector SMADs and SMAD4, which signify active BMP signaling in the cell.